AutoGene. Our main goal is to reverse engineer and turn an Ecoli K12 plasmid into a genetic circuit which anyone can look at to understand the relationship, like activation, inhibition between the genes. Our motivation is that scientists can genetically modify Ecoli K12 plasmid to do what they want, such as glow with GLP genes, but there are no tools which would do the reverse. Turn the Ecoli K12 plasmid into a genetic circuit which they can analyze.
Okay, so basically AutoGene does three things. First, we take the DNA sequence from a plasmid and run it through BLAST to figure out what genes are in it. Then we take those genes and look them up in a database and see how they interact with each other. Like, does one gene turn one or another off? And finally, we take all of that and draw it out as a visual diagram so you can see what's going on. So for our three specifications that we have set for first, that we want the Ecoli K12 gene to be identified with at least 95% accuracy, and then second, we want the parsing into the genetic parts to be at least 70% accurate, and lastly, we want to be able to turn a genetic sequence into a genetic diagram in less than 30 minutes.
So here's our functionality. So in order to turn the genetic sequence into a list of genes which we can analyze, we will first need to find all the open reading frames. This can be done by parsing the sequence to find a start codon, which is ATG, and sound downstream there will be a stop codon, which can be TAA, TAG, or TGA. In the case of which two reading frames at the same stop codon, we will only want to get the best open reading frame, which in our project we have defined that as having the longest sequence of amino acid. Then from the BLAST, it will return us the gene name. The BLAST also returns an identity score for the gene to show how likely it is for the suggestion to be correct. For example, here, step 1 is we will get a clean genetic sequence that only includes the ATCG. Step 2 is identify open reading frames, and then we will translate that into the number of amino acids. This can be done by finding the start codon and stop codon. Step 3 is BLASTp, the open reading frames that we have found. This will tell us the BLAST identity percentage, in which return we will find a protein of it. Then we use this protein to use Swiss process to find what genes encodes it.
Alright, so after getting a list of genes from Chen's part, we will now look up these genes in the EcoCyc database. In the case that we do get a match in terms of genes, we would then find the protein that the gene encodes for, and then we would look up the protein in the EcoCyc database. And inside of that information, it will show you what gene that the protein inhibits or activates, and then using that, we would then create an edge between that protein to the gene. In the case that the gene does not exist in the EcoCyc database, we would then search up that gene inside of UniProt as a backup, just so that we can show the user any information that would be relevant.
The features which I've implemented for users to interact with. First, user can input his genetic sequence either by pasting it into a text box or pasting a FASTA file and click import sequence. This will be sent to the backend, which would compute all the interactions and then return back the result to make a graph that you can see here. I've also added flags for each gene showing how they can be activated and inhibited, and if you hover over any gene on a graph, it'll show you which genes activate or inhibited. If the user feels that the BLAST is being way too strict such that you are not getting any gene at all, they have an option to decrease the confidence score such that genes has a lower confidence score, which will show on the graph. The user can also click on each node to bring you a website showing where all these information is coming from. Another feature which we have implemented is that the user can type the gene name themselves, and it will show the graph out the relationship between the different genes.
Alright, so this is the end-to-end detailed block diagram from the user's input into when the graph is formed. There's two ways in which the users can interact with the software, either by inputting the gene name directly, or they can input the DNA sequence / FASTA file. If the user were to put the DNA sequence or FASTA file, the servers will then read that input. They will parse it and find all of the open reading frames, blast the open reading frames in order to find all of the protein, and then match it to the gene. And then once we have gotten the gene, we would then check that gene on the EcoCyc database. In the case that it does not bring up a match, we would then find it on UniProt, and if that still doesn't bring up a match, then we will identify that we cannot find that gene / protein. If we can find this gene on the EcoCyc database, then we would then get the interactions of the protein to find what gene it activates or inactivates. And then we will package that into a JSON file to send to the front end, which would then display the graph for the user to see. And here's the technical example. This is our take on an oscillating circuit with the FIS, CRP, lacI, and lacZ protein. As you can see here, the FIS and the CRP gene creates an oscillating circuit, and we can use the lacI and the lacZ proteins to check, to measure how this reaction is happening, since lacI does break down lactose.
We were able to create a visual diagram by using the EcoCyc database, which basically has tons of information about E. coli genes interact with each other. And that's how we were able to generate the graph in the first place. For example, lacI and lacZ are common E. coli genes, and EcoCyc told us exactly how they affect each other. The graph will show you how lacI inhibits lacZ, which is true.
And for failure, the EcoCyc database focuses exclusively on genes that occur naturally in E. coli K12. In contrast, genetically engineered plasmid often incorporate genes from other organisms, for example, the GFP gene derived from jellyfish. While we were able to successfully identify these inserted genes and their corresponding proteins, we were not able to further analyze or characterize them beyond that level.
Alright, so this is our web server, and how the user can interact with it is by pasting in a FASTA file. Or by typing in the DNA sequence themselves. So what I'll do here is I'll be putting in the FASTA file, click import sequence, and it'll load. Loading, we can see that plasmid encodes for the protein, or for the gene HLY, BLA, and TET-A. And you can also click on these different genes for you to bring up the website where all these informations were gathered from. Another way in which a user can interact with it would be to type in the protein or the gene name themselves. So first, I'll do lacI, lacZ, CRP, and FIS. And this would create the oscillating circuit that we had shown earlier on the slide. On each of these flags, you can hover to see what gene activate or inactivate it. And you can also again click on all of them to see more information.